Yes, sir. We can see your screen.
Okay, that's good.
Yes, sir. Sir will join within few minutes, sir. Still, students are
that's-
also joining.
Yeah, that's fine. That's fine. I was just checking how it works. I never used it.
Yes, sir.
Hello, Nitin sir. Let me check. Am I audible clearly?
Yes, you are audible, Priya.
Okay, thank you, Nitin sir.
Okay, we still have 5 minutes. How was Valentine's Day there for everybody? No answer?
Respond, dear student. Hello, good morning. Can you see me?
Oh,
Visible.
Hello.
yes. Yes.
Okay, good. Good morning. Good morning from here. Good morning from India.
Yes, this is our good night here.
Yes, for you it is a good night actually. It's time to good night. Yes.
Yes.
So first of all, yes, good morning and thank you very much for accepting our invitation.
Oh, yeah, that's
Yes.
very good.
Now, still there are 5 minutes to go. So maybe around 10 o'clock we'll start. So actually let the people, students and faculty members join, isn't
Okay.
it? So how the things are moving there?
Yeah, let me figure it out how I can see the screen. So, fellas, what you can see there? What do you see on the screen? Do you see everything? I cannot hear you.
Yeah, I can see your video.
No, can you see my slides?
I can hear you.
Okay, so
Yeah,
you
I
can
can
see the slide.
as well.
Slides are visible, sir.
Okay. Let me put this.
Yeah, it's visible, sir.
Okay, good.
सांगा ते फास्ट वर्किंग करते ते.
Yes, Priya.
Sir. Yes,
So
sir.
now let us see. Just move. So more than 70 participants are there. Now actually we can start. Okay, begin.
Sir, am I audible clearly?
Yes, you are very much audible. How about me?
Yes, sir.
Hello, Narendra, sir. Am I audible? Am I audible to you now?
Yes, sir. Yes.
Hello, Narendra, sir. Am I audible to you?
Yes, yes.
Yes. Okay, good.
Can I start, sir?
Yes. Go ahead.
Okay, sir. A very good morning and a very warm welcome to all respected teachers and esteemed speakers and all dear students. This is our 7th online lecture series on advances in life science research under UGC-STRIDE Component 1 and Maharashtra Academy of Sciences Kolhapur chapter, organized jointly by Department of Biochemistry and Department of Microbiology, Shivaji University, Kolhapur. Now, I would request to Professor Dr. K.D. Sonovne, sir, please give us a brief introduction about Dr. Narendra Sankpal, sir, who is our today's esteemed speaker. Over to you, sir.
Okay, good morning, everyone. I hope I am audible to everyone. So thank you. Thank you introduction about this particular lecture series. So on the behalf of Department of Biochemistry, Department of Microbiology and Shivaji University Kolhapur, I welcome Dr. Narendra Sankpal, sir, who is our alumni of Department of Biochemistry. It is a proud moment for us, sir, that you have accepted our invitation and actually to deliver this particular lecture or particular your talk or to talk about your research in the lecture series which we had organized. This particular lecture series had been inaugurated on the 8th February this year and actually by the auspicious hands of our Honorable Vice Chancellor, Professor D.T. Shirkesar. And for this purpose, we had chief guest from the Jahangirnagar University, Bangladesh, Dhaka, Professor Shoyal. So he had also delivered actually very nice talk on the plant biotechnology and applications of plant phytochemicals in order to cure or get rid of certain certain diseases. So with respect to that, this lecture today, which Dr. Narendra Sankpal sir is going to deliver on immunotherapy, current advances and future perspectives. So, sir, welcome in this particular lecture series, and thank you very much for again accepting. So, as a actually, let me introduce Dr. Narendra Sankpal sir. So, as I had said, he is alumni of our Shivaji University Kolhapur and particularly Department of Biochemistry, Shivaji University Kolhapur. Passed out MSc way back in 1993. He had completed basically 1993 as a Bachelor of Science in Chemistry from Shivaji University Kolhapur, and then completed post PG from Master of Science in Biochemistry from again Shivaji University Kolhapur, our alumni, and then started actually joined for PhD in Doctor of Philosophy in Biotechnology from University Pune, and particularly their work area or particular work culture, that is nothing but National Chemical Laboratory Pune, one of the prestigious laboratory in India. That actually I am happy to tell you, students, friends, and faculty members, that basically Dr. Narendra Sankpal, he's actually 1 year senior to me for this master program. And then he had joined initially at the NCL Pune, and then again consequently in the second year I also joined NCL, and then we were having very nice discussion during my PhD, and I got immense discussions about the science, about the biochemistry, about the biotechnology with Dr. Narendra Sankpal sir, and still that is continuing, right? Still I am in touch with Dr. Narendra sir, and we are getting very good benefit, not in the benefit, but we discuss continuously on the science. And recently we had also started working as a collaborative research work. And actually Priyanka, the student who just now started this activity, she's doing work on this particular all right. So about the academic achievements as well as employments of the Dr. Narendra Sankpal sir, he had initially joined as a 2002 as a research associate at University of Virginia Health System, Virginia. Then from 2006 to 2010, he had joined as a staff scientist at Washington University School of Medicine, St. Louis. Then 2010 to 2021, he had worked as a senior scientist at Washington University School of Medicine, again St. Louis. And currently, since from 2021, he is working as assistant professor at Northern Thoracic Institute, St. Joseph Hospital Medical. This is one of the faculty position. He is working at one of the prestigious university that is Norton. So his goals are to work or to understand basically causes, mechanism of the lung cancer, and he had published prestigious his research finding in the prestigious journals, and completed several research projects with respect to this lung cancer. With respect to, I would just like to highlight few things: that role of epicam in EGFR and RAS signaling in breast cancer, breast, lung, as well as GI cancer. Then he had also worked on targeted cancer therapy using epicam, that is SCFE antibodies, to inhibit cell motility in activated RAS files. Likewise, he had completed several research projects. About the honors and awards, initially in 1995, he got a fellow graduate aptitude test in engineering Pune, and he had cleared during his MS itself. Then 1996, got a junior research fellowship from the Department of Science and Technology, New Delhi. Then 2003, got senior research fellowship from again CSIR India. And then 2016, he got a R25 strength program fellow as a cancer genomics and outcome research clinical research training center at the University of Washington University School of Medicine. So he is holding several editorial responsibilities as well, like transplant journals called Transplantation, Development of Dynamics, Cell Biology and Toxicology, Carcinogenesis, International Journal of Cancer, then Journal of Immunology Research, Molecular Cancer Research, one of the prestigious journal that is Molecular Cancer Research. And I could see from his CV that he had also published several research papers in this molecular cancer research. About the professional societies and organizations, he's a active member of American Association of Cancer Research, the American Society of Cell Biology, American Statistical Association, and he had received several grants from the different funding agencies. I would not list out each and everything, but happy to inform you all that Dr. Narendra Sankpal had also got one patent to his credit when he was working at National Chemical Laboratory. Then about the basically he is working in the clinical research with respect to lung cancer and other aspects, and he had also developed quite good research collaborations with actually prestigious scientists or great scientists all over the world, and he had published several research papers, onto which I would like to list out some of the list of journals like Oncotargets, Oncogene, then Molecular Cancer Research, then Immunology, Journal of Surgical Research, right? Cancer Research. These are the prestigious journals that he had published, and also he had also published his one book chapter and presented his research findings in different conferences, national as well as international conferences. So students, dear students, friends, faculty members, we have very eminent faculty, eminent alumni of our Department of Biochemistry, and happy to inform all of that Dr. Narendra Sankpal sir has accepted our invitation from his although busy schedule. And I know that he has recently shifted to the phonics, and he is also having a lot of good work, lot of work to do. And although he's quite busy schedule, sir has accepted our invitation. And now I request Dr. Narendra Sankpal sir to actually deliver his talk. Thank you very much, sir, for in this particular lecture series. Over to you, sir. Narendra.
Yeah, thank you, Dr. Sonane Kailash. That was almost all my CV.
Yes, yes, yes.
Yeah, so we came from a small place, and the only thing we know, education, going ahead, is the only path of success. Show your abilities to help people in need. So I think this was one of the best field we started. So today I will talk about immunotherapy. This subject is very close to me for several reasons because I worked in this field for almost like 7 to 8 years. Washington University is almost one of the first 5 university in US. And for immunology, it's like top 3 in the world. And there is it's very big university, a lot of faculties, 2, 3 hospitals connected, several clinical trials, a lot of several scientists. Even one of those COVID vaccine development was partially done at Washington University. There are 2 or 3 big scientists who work in WashU. They were consultant to Moderna. So to going ahead with the whole talk is about just a brief introduction because this whole subject is such a huge. If you want to publish one paper from start to end, there will be at least 50 authors. So the work starts from usually from patients, taking patients, samples, genomic sequencing, then predictions, computational work. Then it goes to lab, then lab designs, manufacturers. Then it goes back to patient again. Then we analyze patients. So it's a huge process. There are doctors involved. There are nurses involved. There are scientists involved, technicians involved. There are facilities involved. So it's very big contribution. And when everything comes together, you develop a very successful project which can help people. So in my talk today, we'll talk mostly about the overview, how we did it, what are the success rates, what are the features of this immunotherapy, and what actually is involved, and what exactly we can see. So this is a beautiful town, city actually. I moved from St. Louis to Phoenix in October. It's very dry land like Solapur, Sangli, Satara. You can see mountains and lot of cactus. So everywhere there are trees, but this is kind of dry land. I was wandering around. There are a lot of walking trails. Like when we were in Shivaji University, we used to walk on those mountains areas and catch crabs and cook them. I remember those good memories. So I like to go all these areas where we can see different cactuses. Like there's a Sugaro, you can see these are protected plants. And there's a barrel, especially the prickly pear. When we were kids, I remember when we used to walk in the mountains, we used to eat those actually. So it's a beautiful town, big city, a lot of people from South America. So this is the St. Joseph Hospital. It's in the heart of the Phoenix city. It has a neurological institute, several research institutes. Our institute is Northern Thoracic. Mostly it's related to thoracic research. This part. So basically, what is immunotherapy? So yeah, so immune system is made of several organs, including lymph nodes, respiratory system, skin, stomach, spleens, white blood cells. There are mostly the first innate immunity is mostly our skin, cornea, mucous membrane, which protects any outside bacteria, virus, anything which can infect us or make us sick. The acquired immunity is the most interesting because whenever we get anything outside in the form of bacteria, virus, we have a natural gift system. It develops antibodies from T and B cells, and at the end, all these outside organisms are killed to keep us safe. Organs involved, they're like adenoid glands, bone marrow, lymph nodes, lymphatic vessels, spares, patches, spleen, thymus, tonsil. These are just organs. But if you go into the deep, there are so many T cells, B cells, macrophages, NK cells. Okay. Can I move this away? So mostly I'll be talking about treatment in the cancer research. And the most prominent therapy is coming in different institutions where there are facilities is immunotherapy. So usually when our patients are coming with cancer, the first thing is, can it be cured with surgery? Because cancer is a growing part in your body, so it can be just excised out from the body using surgery. Radiation therapies are very common, chemotherapy is common, immunotherapy is building up. For some cancers like breast cancer, ovarian cancer, they use hormone therapy. Targeted therapy is gaining some attention. Also, stem cell therapy is the next therapy in the list. So before going to the immunotherapy, we need to understand exactly how this immune system works, how does it checks or fails. Immune system may be regularly fighting of cancer. So in our body, a lot of people think, or researchers think, we are getting cancer almost every day or maybe every week, but our body fights against it and kill the cancer cells. But sometimes these cancer cells can grow aggressively and they can evade immune system, so they can fool the immune system and hijack the immune system to help cancer cells to grow. So then cancer cells can turn off our body's immune system. Also local system. Sometimes cancer cells use both innate as well as adaptive immune cells to recruit them and grow the cancer cells. So for example, when cancer cells use immature immune cells called myeloid-derived suppressor cells, MDSC cells, they basically deceive the signaling and the cancer cells shift the growth of these cells to help tumors to grow. So basically, in this case, it's at very delicate level to see can this therapy help or really grow the tumors. And at the end of this, immune system can work three different ways. Either it can help tumor growth, it can destroy, or keep the system in the dormant phase so it will grow when your immune system is weak in the later age. Or basically, these cancer cells have a mechanism of escaping the whole immune system, and they can come back later. So from 1960s, this field is in the research, but since there were not many advances in the science like genomic sequencing or a lot of understanding of immunology, this field was kind of on hold for a while. So when sequencing started 10 years before, everybody was interested to develop therapies for patients. And the first thing is, how can we develop it? So for example, immune system recognizes our own genome sequence, our body. Whenever there is a mutation in cells and the cell is moving around in the body, our adaptive immune system can track it down and kill the cells. So that was the main focus, and that is still focus to use that system to kill the cancer cells. So how can immunotherapy help? When cancer cells are able to hide from immune system or block the immune responses, or if it just isn't strong enough to kill the cancer cells, immunotherapy can help to strengthen and restore the immune system's ability, which our body has to fight the cancer. And can it be done? Yes. There are two classical successful examples. One is HPV vaccine, and there is another hepatitis B vaccine. These vaccines are commonly given in US and all other countries, when kids are at in high schools, it's a requirement. Also, some people who has weakened their liver function, doctors might suggest to give hepatitis B vaccine. So these are the examples as a preventative care. The system, the science behind it, it's the same. You have antibodies like a COVID vaccines, and whenever you have infection next time, your beta cells has a memory. They can generate antibodies and they can protect you in the future. Now, what are the types of immunotherapy used in the cancer? The classical examples are monoclonal antibodies, then checkpoint inhibitors, several cytokines, vaccines. We'll talk about this today. And there is a CAR T cell therapy. This is the whole new field. People are interested. There is some success rate in the field, but there are a lot of unknowns and there are a lot of advances, people learning about it. So the best thing about using immune system or immunotherapy is for the personalized therapy. The advantage of this personal immunotherapy, how can I move this? So the advantage of the immunotherapy is because immune system is a mobile. Anywhere in the body, if it could be brain, it could be tonsil, it could be liver, anywhere if there is a fault or if organ has growing cancer cells, immune system patrol them and they can destroy it. The immune system, since being part of our body, it amplifies those T and B cells so it can protect us. Also, immune system can evolve based on the signals they get every day. So once you evolve, you can learn more. So basically you can have more protection. Also, immune system can recognize almost limitless number of targeted molecules. It could be bacteria, different genuses of bacteria or fungi or viruses. And the best thing about the immune system is it has a memory. Now, when we're talking about SARS-CoV virus, a lot of like when I was in WashU, the group was studying what vaccine works better to evoke B cells. So the B cells are made in the bone marrow. And once you are infected with something, the B cells or those memory B cells, they can have a memory. And when you are infected again with viruses or any other disease in future, those cells can make antibodies within a few days instead of like having 2-3 weeks time, and you can protect yourself. So some of the classical examples of antibody is checkpoint blockers and immunostimulants. So there is ilimumab and nivolumab. There is also anti-CD40 and 4-1B. These are the antibody therapies are currently used as a checkpoint blockers or immunostimulants. So for example, of immunotherapy, now the most commonly used are monoclonal antibodies, but there are research, people are using fusion proteins, CAR T cells, dendritic cells. So you can take the cells out, change them, and put it back again. So CAR T technology is very interesting. They take out your cells from your blood, they genetically change them, amplify them, and put back again in the patients as a custom therapy. So the lymphocytes have similar function than T or T cells, cytokines, use of cytokines, oncolytic viruses, bacteria. These can be all used in the immunotherapy. Now, when we talk about vaccines, the vaccine is nothing but it's kind of preventative care, but also it can be used as a treatment platform. So vaccine can be used as a whole soul viral vector. Now we can hear many times about SARS-CoV that there is adenovirus. It was partially killed and it spike protein from the from the virus can be used as a vaccine. People can use the DNA vaccine. Currently, there is a company called Genios. We worked with this company for 3 years when I was in previous institute. RNA vaccines, we know Moderna has RNA vaccine. Protein vaccines have some success rate. Also, killed or attenuated virus in formaldehyde can be used because when they're attenuated, the virus structure stays same, only it cannot amplify. So the extracellular proteins can be used as a source of antigen. So these are the traditional approaches, protein and killed viruses. So for example, if you take flu virus, most of the flu virus in the world these days, they are still made with attenuated virus. They amplify virus in the eggs, they attenuate it with formaldehyde, and they just inject it back to the patients. But the new viral vectors, DNA viral vectors were controversial for a few years. But since COVID, viral vectors are getting, especially adeno-associated virus, they're becoming very famous. There are several reasons they can be amplified, they can be manipulated, they are safe. The only some disadvantages are they can generate immune response. So people have to change them, design them the way they want so they can have a minimum immune response. The DNA vaccines, we worked on this vaccine for several years. They're very cost effective. So because you can just make a plasmid DNA into the lab and you can use for animal studies. RNA is very new field because you have to take the plasmid, you have to reverse transcribe the RNA, or you can just use polymerase to amplify the RNA and change the ends of the RNA, make them stable, use lipid nanoparticles, they call LNPs, where RNA is stabilized, and it can be used as a vaccine. Now, Moderna in US is using same technology. Pfizer also using same technology. Proteins are kind of expensive because protein making is not just expensive, but also there are a lot of checks and dues involved because you have to design the DNA, make the protein, purify the protein. There are yields, there is a purity, stability, also shelf life. So compared to all this, DNA vaccine is very easy to make for studies, and at large scale, then you can switch to RNA or protein. Now, when we say DNA vaccine, how does the DNA vaccine work? So DNA vaccines are nothing but just like a plasmid we use in the lab to transfect any DNA or transform the bacteria. It has different plasmid amplifying related DNA sequences. You can clone your own gene or anything in front of after the promoter, and you can express the DNA, that gene. So for example, here, some of our lab, they use the old style we used to call gene gun, but now this is the modified version for the patients. So basically, you amplify the plasmid DNA, pack into the vial, and there is argon or helium gas. So based on the pressure, all those DNA, they are injected under the muscles of the patients. When the DNA go to the muscles, DNA makes RNA, RNA makes protein, and that protein our body identifies as antigen, and those antigens can help to evoke our immune system, and that's how you are protected from the future attack of these kind of antigen-related organisms. Now, the DNA vaccines, they have several advantages over other vaccine types because they can stimulate both adaptive as well as say, both immune systems. You can genetically modify easily. Then DNA encoding tumor-associated antigen TAA can be also designed to induce CTL responses for the cancerous cells. These DNA vaccines are low immunogenic because there is nothing much in the plasmid itself except kanamycin or neomycin gene, other than the gene of interest we use. So this in vivo, we worked with this company for 3 years. I was one of the main person involved from design to manufacture to quality control And then later somebody else took over. So when I work with this company, we have a lot of good experience, like how working in the university versus working for a company, there's a huge difference. We are very casual when we work in the lab. Oh, it's fine. But when you work with the company, they have deadlines, they have quality controls, they have meetings. So basically, I was exposed and I was very fortunate to work with this company and their staff from design to execute. So this is one of their paper they show using DNA vaccines. You can use this for cancer treatment. So basically, they designed uh, for some patients. So basically, there can be two type of therapy on any immunotherapy. One is personalized, and one you can call off-shelf. Means if somebody has like a flu-like symptoms, somebody has a similar type of cancer like P53 mutated, KRAS, it's kind of common cancer, but somebody might have different mutations. So like all the researchers now, they're working on two different options. One is personalized therapy. So you can sequence the genome, find out the fault, see if that gene can be used as a immunogenic, clone that sequence and put back into the patients. Now your hindered immunity is evoked based on the doses of the extra gene. For on shelf, basically you can just develop the common therapeutics like we take any medicine for fever. So these are the two things all these companies now are targeting. So what is the theory of this DNA vaccines? Like when we say, oh, it's very easy, but when we say it's easy for like reading, writing, but actually when you go to the lab and you start from scratch design, there are so many committees, there are so many scientists, there are so many things are involved. It's like a team effort. So for example, we design the plasmid, then we get it quality control, we do fermentation, then we just do plasmid prep, then purified plasmid can be used as a DNA vaccine. And now once we have the DNA vaccine, you cannot just eat it or drink it. It has to go in your part of the body where the gene can be expressed. So for example, if you take the DNA and inject, so the question comes, what area of the muscle you can inject or where you can inject. So basically people by experience in animal works, pigs, or non-human primates, they optimize. Okay, if it is like it can be for some animals or some area, it can be intramuscular, intranodal, subcutaneous, intravenous, or intradermal. And based on these needs, they also have developed different electroporation or just simple machines so they can inject. So when it is done, then you can you have evoke the immune system. MHC class 1, 2 based immunity is activated. And basically the cancer cells can be cured. So now overall, how this neoantigen DNA vaccine manufacturing process works. So the first part we discussed was about just like what is the immunotherapy, how it can be used. The second part of portion will be exactly what is involved in this whole process from patient to going back to the patient. So usually first step is selection of the neoantigen and the vaccine design. So the first process is whenever the patient comes, there is institutional protocol. We basically take several patients, take their samples. We send those samples to uh, uh, doctor's office. They check the tumor type, tumor confirmation. They send sample of it to the IHC or the tumor core. From tumor core, they confirm it is tumor. Then it goes to the genome sequencing. In genome sequencing, they take the sample, they QC. Many times the samples fail because of the quality, amount. Once it is done, then the exomes are sequenced, there are some standard probes involved. So we don't sequence the whole genome. It's not necessary. We know like there are certain genes, maybe like 5,000 genes are commonly mutated in the cancer. So we just sequence those. And so once they're sequenced, some sequences are recurring, means they occur again and again. Some sequences, they're rare, very few times come. Some sequences are new. So based on those sequences, that data is generated and sent back to a committee where the committee decides, okay, can we use this gene or not? Can it be toxic if we put it back, or can it have any side effect? Then we send it back to multiple softwares where based on MHC class 1 or 2, it is decided if that protein where it is mutated, can it be antigenic? Once it is done, then that part comes to us. We design based on our experience. For example, if the plasmid DNA has 1 kb sequence, 1 kb can express, like suppose 300 amino acids. In those 300 amino acids, what we do is we put a ubiquitin sequence, then we put 12 to 15 peptides for each gene. Maybe we kind of shuffle a little bit. So each plasmid can have at least 20 to 30 epitopes from different genes. The company we worked, they used to do like 30 to 40 epitopes. So each epitope is anywhere between 12 to 15 amino acids. So your DNA promoter starts amplifying ubiquitin epitope 1, space epitope 2, epitope 3. So at the end, when the DNA is injected into the patient, the ubiquitin kind of ubiquitination of the gene breaks all those epitopes into small peptides. And those peptides are used as in an antigen presenting system in MHC class 1 and 2, and they're treated as an antigen. And then our system wakes up and start making more antibodies or more strong immune system. So that was basically the theory in the first. So once it is done, then at the end, we design the plasmid, we send it to a company. The company clones the whole gene, they send back to us. We do a QC, then it goes to the manufacturing practice. So most of the universities, they have their own small GMP facility. Basically, it's a small production unit, very clean, all monitored. FDA or regulatory people, they visit there to see everything is going fine, clean, no problem. So once it is done, then it's a 1 week process. We transform the DNA, we culture, lyse, elute, purify, and then there are like almost 12 to 13 different tests we do on the quality of the DNA. And at the end, it goes to the further testing in the lab. I can show you the next slide. So after that, simultaneously, it goes to next lab. They formulate with some adjuvants like IL-2 or IL-12 or something else, and they just send it to the pharmacy. But in the meantime, when it comes to the lab, we do several tests. Like, for example, we do plasmid activity. For example, if you design the vaccine, you should make sure it expresses. So we transfect, we take the RNA, make cDNA, do qPCR, it should pass expression level. Also check the plasmid homogeneity, because once you make it's alkaline process, so you are exposing the nucleic acid to very strong alkali and very strong acid. So we should check the homogeneity if it is like full or partially nicked or circularized. We also check contaminating RNA if there is any bacterial chromosome or DNA. We sequence the full plasmid 2 times, 1x or 2x, to see if there is any additional mutations, because this is like a medicine, it goes to the patient. If there is any mutation, something else can happen instead of treatment. Then there are some small like appearance, pH, concentration, purity, since this plasmid comes from bacteria, bacteria has a lot of LPS, and those LPS can work as endotoxin. So it can have more dramatic effect than your immunotherapy if we have basically a lot of endotoxins. Then at the end, we do the sterility test since it was supposed to be toxin free. So this is how we start and end. And at the end, when everything is done, then this DNA goes to the phase 1 or phase 2 trials. Like our lab is involved. Our lab was one of the first in US. Like 10 years before when I started there, I was not part of the group working on this part. But they published that, okay, breast cancer, this gene is this therapy, immunotherapy can be very safe. Before that, there was no safety issue. Everybody was kind of scared. Can we do this in people? People have done in the animals or small animals, but that was first trial. We came in the news and it was we got several grants after that. So this is the most recent paper we published last year. It's not published yet, it's still in the matrix, but I was part of it. Basically, I was involved into the design, manufacturing, QC, and I was monitoring everything except from the patient when it goes to the patient. So this paper shows how new DNA vaccines can be safe, feasible, and capable of inducing immune responses in the patient which has triple negative breast cancer. Now, if you take a breast cancer, there are kind of 5 different types: luminal, AB, HER2, then there is basal A, basal B. These are the category based on the gene expressions. And since understanding gene expression is very important into the immunotherapy. Triple negatives are very aggressive breast cancers. Mostly it happens with the people with the stress or certain Asian Black community. And it happens in later age. And these cancers are so aggressive. People usually they are detected kind of late, and the treatment hardly works on these patients. But since this triple negative breast cancer has a lot of antigens or immunogens from cancer, they're very interesting to treat these patients. So that's why our group started with this. Immunotherapy has good success in melanoma, skin cancer, partially with brain cancer, glioblastoma, and the third part is like triple negative breast cancers. So this trial was going on for almost 5 years. At the end, we showed, okay, it works, it's a safe, and the life of the patient can be improved. Instead of like 2 years, now the patients can live 4 or 5 years or maybe more. So we discussed about a little bit about immunotherapy. We discussed how we produce in the lab, how it goes to the patient. Now, when the trial starts, there's a different group of clinicians and nurses. Then we have to start enrolling patients. So for this trial, we started with 35 patients. So in India, it's very easy, but here it's very difficult. People withdraw, they say yes, no, they want money. So initially we started with 35 patients. So from 35, 6 patients have complete response. They said yes. 1 patient was DCIS, advanced cancer. 3 patients has insufficient tumor, a sample for DNA sequencing. 4 patient withdraw, 3 had disease progression. So basically, when you select patients for treatment, you should have some kind of criteria. The criteria we have is, okay, first they agree, then you can remove some of the patients if they have aggressive cancer. They should not have autoimmune disease. Even if we get enough samples, sometimes transport, freezing, thawing, it kind of destroys the samples. At the end, we treated 18 patients. So the treatment regime was like this. You take the biopsy, of the patient who has been initially gone through non-adjuvant chemotherapy. Can you see my cursor here? Hello? So they go through non-adjuvant chemotherapy, okay, or surgery, and then basically all these patients, they have such aggressive cancer, we cannot do trial medicines on these patients. So these patients go through basically chemotherapy or radiotherapy, and after that, they come to us and we started treating them. So in the reason we call adjuvant therapy, because having just DNA vaccine is not enough. So we use IL-2, IL-12 or something else as an adjuvant. And the dose goes to like week first, day 1, day 5, 9, 11. So every 2 or 3 weeks, we give at least 4 milligram of DNA vaccine to the patients. And then we withdraw their blood sample and then we freeze that. So if when a patient has 3 vaccinations, we take the samples before and after control as well as treated, and we keep those for later analysis. Once it is done, the same time, we also sequence the whole genome. We design vectors and then manufacture in this first phase. So now, when you designed, you introduce your vaccine to the patients. Now, what you can do next? So there are so many criteria we have to look for. First thing is whatever you injected, is the patient getting immune response? If it is getting, can you confirm that in vitro into the lab? So yes. So this graph shows, the first figure shows all these different, for example, these were the common genes mutated. We use those as antigen into these different patients. If you can see figure B, these are the different patient codes, and these are the mutated genes, and we can categorize them based on either what kind of mutations those are, can we use them, are they immunogenic? So once this is decided, then we design, and then we go to the next level. So the very critical portion of this immunotherapy, it still lags a lot of details, is how you design the immunogen or neoantigen. So these antigens, as we know, these are small peptides, 12 to 15 amino acids. So for example, if we take figure A, ZNF646, this is the name of the gene, and this is the sequence. The H shows the mutation. Now the question comes, should you keep the mutated amino acid at the center or shift a little bit here or there? If you shift a little bit right, your left side is kind of have a amino acid sequence, which is immunogenic. So it's kind of perfect. If you move on the right, you miss that. So basically you have to do some trial and error, design 1 or 2 epitopes for each mutation. Sometimes that mutation on software, it shows it's a highly immunogenic on class 1, class 2 MHC, but sometimes it turns out to be totally nothing. So similarly, what we did was we just shifted these epitopes on the left and right, and we read the data before and after vaccination. So this is basically an assay called ELISpot. You can see the response of the peptide based on the immune modulatory system. So similarly, you can see all these, we tested all these epitopes for their in vitro activity testing of the patient samples. So you can see pre-vax, post-vax, there is a response. So for example, if you see post-vax, we can see very good on OP1, but not others. So similarly here, pre-vax, you can see not good response before, but after we can see those good dots. So this is the first criteria. Since people are building the database on this, also they say, okay, if OP1 peptide works, next time when there is a mutation in this gene, we can use the same. Hello? So now how we can do the next stage. So new antigen induce antigen specific immune response. So for example, then the next we tested, can we develop an assay to test these samples we get from a patient? So what we do is, whatever epitopes we designed for the treatment regime, we also synthesize the same peptides, and then we tested into the lab. What we did was we took the PBMCs from the patients before treatment, and now we know we already treated them. So we use those PBMCs with these peptides, and we can check in the lab, whatever we treating them, can we see same response at the lab? So this data shows we can test those different peptides based on from the patient's PBMCs, and you can predict what is the outcome will be for that patient. If the patient has good immune response in vitro studies in the lab, that means we can expect better survival of that patient after treatment. Now, the next question comes, it's the clonal variability. So for example, we treat the patients with 5-6 different gene epitopes as an antigen, and only some of those works very efficiently. So this data shows, for example, if you see B and D, it shows only one clonal variation is more effective than the other peptides or other epitopes. Because whenever we do these experiments, there are a lot of question comes, okay, exactly what peptide work? Or does like all peptide work? Or just one or two has partial efficiency, or others, they're just like a dead. So this data shows you can also do those studies in the lab and predict patients' treatment regime in the future. So this is one of the last figure when we publish. So at the end, this is called survival curve. So in the survival curve, your data should show exactly what you treated, did it really work? So your goal is the patient, you can see this patient has a disease or cancer progression. The patient might die within, say, 20 months. Now, your treatment regime, can you increase the life of the patients more than 20 months? So here, what we show, like A is vaccinated during their cancer progression, and B is the control. Now, if we see this VVS control, the patient survival rate is almost like 85%. Compared to here, it's almost like a 49%. The statistical value, p-value, is very strong. So this shows when you select patients carefully based on their genotype or availability of the antigens, you can use this immunotherapy very successfully, and you can increase the patient survival rate drastically, almost 2 times, based on this study. Now, the patients we enrolled, they were coded like this, different code names, their age, race, stage of the tumor, then where their treatment was with neoadjuvant or adjuvant. So all those details are here. How many vaccine epitopes were designed per patients? Do we see immune response in the lab versus after treatment? And some of this data we can see immune response was yes, except these 2 patients. So this shows, if we use the DNA vaccine, it's very easy to get the immune response. Now, when you do clinical trials, there are a lot of restrictions because you're dealing with the people and their lives. So every drug you test in animals, there are certain criteria to see what happens to people as a side effect. So example, like a fever, chills, fatigue, any mental disorder, CNS, cardiovascular, pulmonary, anything else, metabolism. So all these you have to score based on the guidelines. If it is 1, that means it's kind of marginal. If it is 5, it's not acceptable. All in the whole treatment, it shows all the side effects very, very marginal. And most of if you take any medicine, there are also some side effects. But since these treatments are based on the immunotherapy, you're kind of modulating the immune system of the person. You will have some side effects, but usually these are very moderate and acceptable. Now, this immunotherapy has a very bright future. Most of the universities, like for example, Washington University is one of them, Harvard, Dana-Farber, MKCC. These universities are at the front, including Johns Hopkins, doing these studies because they have facilities, they have hospitals attached, and they have very good scientists who know immune system. And usually all these studies, they need very good collaboration between institutes. So for example, the Genios company who worked with us for breast cancer, same time they were working in Johns Hopkins for glioblastoma in brain cancer. Same time they were working in Australia for something else. So basically, big companies, pharma companies, big scientists, scientific groups, when we don't have enough breast cancer patients like previously, so we got some of the samples from Dana-Farber, and basically we send our treatment regime to them by mail so they can get treatment there. So basically these kind of collaboration, science, networking is very important to do these efforts as a treatment the future. So all this immunotherapy, the key point still is epitope design predictions. So the database is building, science is building, and this portion is still very critical, but there are advances going on in this sphere. Tumor heterogeneity. So this is very complex thing because what happens whenever there is a tumor or cancer in the body, it keeps growing, but also the number of the mutations keeps growing as the tumor grows, keep growing. If you see there are like over 10 mutations in first year, and if the same patient, if you sequence the tumor after 10 years, there'll be probably 10 times more mutations. So this tumor heterogeneity basically is one of the major challenge in cancer treatment because initially there was one tumor type, one cell type, but after 4 years, 5 years, when the tumors comes back after treatment, it has a lot of different tumor mutations or different tumor heterogeneity. They call clonal variation in the cancer. And to treat those cancer is very difficult. This kind of clonal variation is very common in lung cancer or pancreatic cancer. So that's why these cancers are very difficult to treat compared to breast cancers or glioblastoma. The next thing is understanding cancer and immune systems. So research is going on every day. We learn something new from immune response or cancer, this gene, that pathway, this new drug. The next thing is delivery. So whenever there is a drug, small molecules, chemicals, they're very easy. Either you can just take orally or you can inject or go through IV. But all these DNA, RNA platform, there are still delivery options very weak. For example, when we transfect DNA, the amount of signal we get 1/10 of that signal from the patient. So that means when we deliver 4 milligram of plasmid, we're not getting that much response. So a lot of companies, also universities, are working how to deliver. So for example, for this study, we use a tri-grid electroporation unit. And now we can hear there is something else coming up, which is more efficient. So basically, when you design a study, you go through IRB, FDA, you cannot change anything right in the between, but we're planning to use the new machine next time. All this study now, as we see, it can be as a personal because each cancer is mostly like 99% personal. Rarely, a few people can have same type of cancer clonal similarity. The third thing is we have to use invasive process to take the patient sample. The next thing people are working on liquid biopsy means you can just take a blood from a sample, and can you use that information to check the mutations or like circulating tumor cells as a liquid for liquid biopsy, or you can take some LcRNA or circulating tumor DNA, some circulating cells, which you can use as a single cell sequencing resource. So there are a lot of new things going on in this field to help. So at the end, can we say cancer vaccines can generate and amplify tumor-specific immune response? Yes, it does. And there are several other studies going on, not just in our institute, but other institutes also in some Europe. The data is mixed sense. The one thing is the only reason some people say if you treat the patients now and if you treat the same patients again, second time, can you have the same response? And the answer is kind of no, because everybody has different immune system, age, race, sex, everything is different. So basically all this learning process is going, there is a database building up, but at the end, once everything is known, then it will be like very confident to say, yes, we can do it. The immunotherapy, the cost-wise, there are a lot of factors involved. Initially, like 10 years before, we don't have option. We don't have that facility to sequence the genome in 2 weeks. Now we can sequence the genome in 2 weeks. The cost is almost like 100, like in $1,500 to $2,000, we can sequence the exome DNA we want. So that option is coming very clear. After 2, 4, 5 years, the sequencing cost will be further down, speed will be further down. They are developing a lot of softwares in sequencing centers, so everything will be speeding up. So currently, if we get a patient sample, the whole process, design, amplifying, making vaccine, it takes almost 3 months. And you can imagine in the 3 months, the tumor is growing in the patient. So at the end, we're planning to cut this time to maybe like 1 month because you cannot do less than 1 month. In our facility, when we manufacture DNA, we need at least 3 weeks to design, start, clone, manufacture, QC. So at the end, but when you reach that point, based on the scientific information or the new developments, this therapy can have very good future because it has less side effect. You're doing nothing, but you're just tickling your body to grow up the immune system and kill the foreign bodies, like it can be bacteria, virus, or a cancer in your body. So at the end, I would like to acknowledge my center, NTA, Dignity Health. We have hospital. My previous place, Washu, and a lot of people, patients, contributing people, scientists. Thank you. Any questions?
Dear students, now the topic is open for discussion. Do you have any questions? Yes, the students and participants from all the Maharashtra, all over the Maharashtra, Shivaji University, have any questions? You can ask or just type in the chat box also. I think no.
So go ahead.
Supriya, go ahead.
Am I audible, Nikhil sir?
Yes, yes.
याच्यामध्ये बघा चॅट बॉक्स बघा क्वेश्चन चॅट बॉक्स मध्ये
नाही sir, नाही.
No question, sir, in chat box. Okay, now I request to Dr. Nadab sir, please give us vote of thanks.
Am I audible?
Yes, sir, you are audible.
Okay, Priya. Very good morning to all. Our today's respected speaker, alumni of Biochemistry Department, Dr. Narendra Sankpal sir, chairman of this event, respected Professor J.P. Jadhav madam, Head Department of Biochemistry, and Professor Dr. K.D. Sonone, sir, Head Department of Microbiology, convener of this event, respected Dr. Padma Danke Madam, Dr. Shailesh sir, faculty members of Biochemistry Department, Dr. P.M. Gurav sir, Dr. Pankaj Pawar sir, Sandeep Kale sir, student, research students, and teachers. Myself, Dr. Narendra Sankpal, Department of Microbiology, IND. It's my pleasure to have been asked for propose a vote of thanks for this occasion. On behalf of organizers, Advances in Life Science Research ALSR 2022, which is organized by Department of Biochemistry, Microbiology under the UGC-STRIDE component 1 Maharashtra Academy of Science, Kolhapur chapter, and from entire team of ALSR 2022, let me call it's a fraternity of to have together. And on behalf and from my all team of this event, I extend a very very hearty vote of thanks to our today's eminent speaker, Dr. Narendra Sankpal sir, for gracing your important work, sharing with us your finding and opinion today. Thank you for giving us excellent coverage of cancer biology and role of immunotherapy in the treatment of cancer, different cancer, of course. I wish to express my gratitude, you sir, because providing this all kind of information will encourage our student and research student to work in this area. I like to express my sincere thanks to our respected Honorable Vice Chancellor, sir, Professor Dr. D.D. Shirke, sir, Pro Vice Chancellor, Professor P.S. Patil, sir, from Shivaji University, Kolhapur, for granting us a permission to conduct this online event. I am very much thankful to Team ALSR 2020 for conducting these events very smoothly. And at last but not least, from all, I am very much thankful to all audience to be here and listen us very carefully. Thank you very much. Thank you very much, sir, to be here. And thank you very much, one and all.
All right, thank you,
Over to you,
Narendra
Priya.
sir.
Thank you. Thank you.
Hello. Thank you very much for your time and for this valuable talk.
Sure, no problem. Is this available on like YouTube or something? You can
Yes,
hear?
yes, sir. I will send the link on. Already, I have shared the link on the group. There is a live streaming is going on. After 24 hours, it will be available in the departmental profile,
Okay, okay.
YouTube channel.
Okay, maybe you can send me the link.
Yes, sir.
Thank you very
Okay.
Okay, good
thank
night.
you.
Good night.
Thank you,
Yes.
sir.
For you. Yes,
Bye.
yes. Okay.
Good night, sir.
Okay.